We'll start our industry presentations. Again, each of the presenters has been told about the time. The time is listed here. This is just important so that we move things along and everybody's fair to one another. So with no further ado, I will ask Laura Cavanda to come up. Laura's with Astellas, and I think our AV person is going to substitute your slides in there, okay?
Okay, thank you.
Give us a second.
All right.
Okay.
So while we're waiting, thank you to the MSG. I still call you MSG even though you have a little extra letters after, but I appreciate the opportunity to present today. I am happy to see I have 15 minutes, but I'm not sure I need 15 minutes. We have a skimmed down legal presentation for you. But I will add to it at the end so you guys get a little bit more information then. Do I have a? Okay. So I think most of you know who I am. I've been at Astellas for quite a while, and I work in the Global Development Group and have been developing drugs with Astellas since the time that I started there, about almost 21 years ago. And I've been coming to the MSG meeting for about those many years, so there you go. That's how old I am. So Astellas is the combination of two Japanese pharmaceutical companies, Fujisawa and Yamanouchi, and we were founded in April of 2005. We have over 16,000 employees worldwide and 2,800 just in the US. Our primary areas of focus globally include neurology, pain, diabetes, oncology, urology, immunology, and most importantly, infectious disease. In the US, it's basically identical, except we don't really have a focus for neurology and pain in the US in our marketed areas. So I'll spend a couple of minutes here. Most of the people in the room have contributed tremendously to the approval, the studies that led to approval of the 3 drugs listed on the screen. So I thank everybody for all the hard work and participation in all the studies that we've done and contributions. My hat's off to you. Ambisome has been around for a while, so I won't spend much time on that, but you're all well aware of that compound. Mycamine has been on the market since 2005 in the US, and I think it's pretty much available globally now. We're currently still working on some development activities, which include primarily neonatal candidiasis. For Cresemba, which is our newest antifungal agent, which was approved in 2015 in the US, Cresemba is on the market for invasive aspergillosis and invasive mucormycosis. Most of the development for aspergillus and mucormycosis is complete, except for pediatrics. So we are currently working on our pediatric development. We started a PK study last year, and we've nearly completed it. It's still ongoing. And we hope to present data next year. You've probably seen posters that have been presented at ECCMID that have described our PK analysis, the POP PK and simulation, which derived the dose for our PK study that was presented this year. And we also have a poster next year that adds to that data, or next week at ID Week, that describes the dose we took to the clinic. We also have a large registry study that is ongoing that a lot of participants are here, and that continues. We're hoping to close that out shortly. And that's about it. I'm happy to take any questions. always looking for new development ideas.
You get a special prize.
What?
You get a special prize for giving us 12 extra minutes. Does anybody have any questions for Laura? I will say, Laura has been one of the most valuable and committed and loyal supporters that the MSG, under whatever name, has had, and we've always enjoyed our interactions with her. She's been terrific. Thank you. Thank you. Who's next? Who's next? Okay. Okay. All right. Gareth? Whoop. Our next speaker is Gareth Lewis. He's with Maine Pharma. Again, relatively new to the U.S., so I'd like to hear what you have to say as well.
Thank you, Peter. Good evening, ladies and gentlemen. Thank you to MSG for having us. It's a great pleasure to be here in Montana. This is my first MSG meeting, my first time in Montana, and you can probably tell from my accent I'm not round from these parts. It's great to be here, and look forward to meeting you throughout the week. In the few minutes I've got here, I'm just going to give you a bit of background about Maine Pharma as to who we are and really why we're here, and to give you a bit of a flavour as to what we're trying to contribute to this scientific area. Sorry, this would probably help. So first of all, just a bit of a company history. So I'm really proud of this. I might go on for a few minutes, but I'll try to keep it short. So some of you might know Maine Pharma because we've been through a couple of iterations over the years. But really, for me, what's pretty neat is that our company goes back to 1845, which is before I think all of us were born. So when there's a compounding pharmacist and an apothecary who set up a little formulary shop in the centre of Adelaide in South Australia. And he turned that little shop into a family business that went for years and years and years. And so we trace our origins all the way back there, which I think is pretty cool. Now that company grew and grew and grew through the 19th and then the 20th century. And it got to the point into the 1980s and 1990s that it started to really diversify and build into a substantial sort of pharmaceutical company. Now, through that time, it started to specialise in some complex solid oral delivery, and really that's been the common theme as to from 30, 40 years ago to where we are today. Now, as we've grown, then of course that attracts M&A. I think most people around here have been through M&A cycles at some point. It led to the company having a really good injectable portfolio. That led to then Hospira buying out the company, really on the base of that portfolio. Now, as with usual with M&A, there's an acquisition, and shortly after there's a divestiture. So Hospira kept the injectable stuff, and with so many hospitalists here in the room, I'm sure you've possibly handled or prescribed and used some of those products at some point. But then what was divested back out was the underlying solid oral dosage form business. Now, that was a lot smaller, and based in Australia, and so since 2009, we've really started to rebuild the business and regrow to the point where we're now this Australian-American hybrid, and really sort of regrowing and investing in the company. So there's a whole lot of numbers up here on the page. I'm not going to go through everything, but this really just give you a flavour of where we're at now, today in 2018. For me, kind of the most important thing to call out here is our commitment to R&D. It's clearly important for us to be bringing more products through the pipeline. As we grow, as our revenues grow, then so does our spend in R&D. And really, just to emphasise, a lot of what we're working on are these sort of complex oral solid dosage form products. As well as our direct presence in Australia and the US, all of these countries in blue, there's certainly no political connotation with the red and blue here, need to maybe change that. The red and other colours are blue countries are where we have distribution partnerships and other companies helping us to take our both generic and brand products out to other patients and markets around the world. In the US, there's three really components to our business. There's a generics platform, a contract services platform, and a specialty brands. Now, again, many people in the room may have come across some of our dermatology products along the way. But really, the focus of what I'm talking to you about is our growth out into the infectious disease therapeutic area. So this is a step out for us. We're setting up a new business and a new team to be taking this out into the markets. And underlying this is a reformulation of itraconazole, which I'll expand on more in a sec, but that's what's taken us into this new therapeutic area. I'm going to skip this, but really, because the point is then, what is this new formulation of itraconazole? Well, back to my point that we've always been a drug delivery company. Now, assuming that there aren't too many solid form dosage fanatics out there, I'm not going to talk about the electron microscopy, as interesting as it is. The point is, what does it do? So the design of this formulation is just to really essentially modify the release of the drug through different pH conditions. Now, as you all well aware, itraconazole can be challenged by gastric pH. So the design here is to modify that into more of an intestinal pH release and hopefully get to a better absorption profile for the patients. This formulation we're now very pleased to be working with the MSGERC. This is in study MSG15, which we will hear about hopefully more during the week. It's now up and enrolling. We have patients, so that's super exciting. We have a poster up this week. Myself and my colleagues over at the table who are currently drinking wine are looking forward to meeting with you and getting to know you through the week. So thanks for your attention. That's me.
Thank you, Gareth. And I would emphasize that Maine has been extraordinarily supportive of the MSG, and so the MSG15 is the first study we've done in the endemic mycoses in over 2 decades. And I really want to congratulate them and give them our support for supporting something that others have kind of steered away from. But this is the right drug for the right indication, and so it's a really, I think, a good mix, and we appreciate that. Thank you very much. Thank you. Angulo from SCYNERGIA will be giving the next presentation. Thank you.
Thank you, Peter. Thank you, everyone, for the invitation. And really, my intention during the next 10 minutes is really to give you a snapshot of SCY-078, or ibrexafungerp. And certainly, I think that for many of you, it's the first time that you see the name ibrexafungerp. And I don't know if, I think that it was Oliver who told me. I don't know if you know the story, but the genetic name is actually assigned by the WHO. So they assign us this name, and I think that Oliver told me, can you sue them? Because of the name. No, I don't think that we can sue them. But however, it has its advantages. It is, the most important part of the name is really the stem. So the second part of the name, which is fungerp. Fungerp now is the ending of a new family of compounds. So by giving us a totally different name that doesn't end in fungin or it doesn't end in azole, the WHO has recognized that this is a totally new family of compounds. So now we have the fungerp family, and ibrexafungerp is the first of that particular family. We call it ibrexa, so don't feel bad about kind of a shortening the name when you're doing presentations. This is ibrexa for the friends. Okay. A quick summary of the attributes. Many of you have contributed and worked with us in really developing this particular compound. It's a glucan synthase inhibitor, however, has a structure that is entirely different to the known glucan synthase inhibitors, which are the echinocandins. It has somehow a different drug-enzyme interaction that gives it a different properties. In particularly, is less affected by mutation in the most common FKS mutation, It has oral bioavailability, which is obviously quite different from the echinocandins. The activity, it has activity against Candida, Aspergillosis, and Pneumocystis. It has activity against azole-resistant and most of the echinocandin-resistant strains as well. It has an enhanced activity in acidic pH, which is particularly relevant for one of the indications that we are developing, which is VVC, oral and IV formulations. And it has low risk for drug-drug interactions and very extensive tissue distribution. The volume of distribution is about 8 liters per kilogram, which is substantially higher to many of the antifungal agents that we currently know, and particularly higher than the candins. Where are we developing this product on? We're developing several indications. Considering the attributes of the product, we're developing the product in vulvovaginal candidiasis. We recently completed our phase 2b study. We selected the dose that we're moving into phase 3, and the phase 3 is starting at the end of this year. We're going to be developing both indications, acute VVC and prevention of recurrence of VVC. We are also developing an invasive aspergillosis, and I'm going to be talking a little bit more about that. We are starting a phase 2 study as a combination therapy with voriconazole. We are developing the product also in refractory invasive fungal diseases. We have 2 phase 3 studies ongoing, one that is called Fury and CARE, so I'm going to talk a little bit about those later on. And we are also planning to develop the product in invasive candidiasis. We have completed a phase 2A study, and we're in the process of wrapping up our plans for subsequent stages of development. Additional indications were obvious, being an oral glucan synthase inhibitor, chronic fungal infections like chronic aspergillosis, and prophylaxis could be potential future indications. I'm not going to talk too much about the mechanism of action. You're extraordinarily familiar with this. We are a glucan synthase inhibitor, and just to stress that it's structurally different than the candins, and for that reason, we may be binding to that enzyme in a slightly different way. So a little bit what we're doing about Ibrexa in aspergillosis. What are we doing there? First, give you a little bit of the basics, and really this information has been generated by people in this audience, all of that. We do have activity, in vitro activity against Aspergillus species, as you can see here with MAC50s and MAC90s, typical of glucan synthase inhibitors. We have conducted animal models in which we have identified that Ibrexa has activity in animal models of invasive aspergillosis by itself. And subsequently, we recently developed as well the data regarding the activity of Ibrexa when given in combination with an azole. In this particular study, it's a neutropenic rabbit invasive pulmonary aspergillosis model in which we observe a very noticeable synergistic in vivo activity when the Ibrexa fungal was combined with isavuconazole, either by really decreasing substantially the pulmonary infarct score in this particular model, which is indicative of really the tissue damage, increasing substantially the survival, and very interestingly, really decreasing the galactomannan antigenemia. All this information had led us to really envision what we could do with this particular product for this indication. And right now we are starting a phase 2 study in invasive pulmonary aspergillosis, in which we're going to be comparing really the activity of voriconazole as a monotherapy versus voriconazole given in combination with Ibrexa fungerp. We are in the process of really selecting sites at this point. This is a proof of concept study, about 60 patients, 30 in each arm. And really the purpose of this study is to better understand what are the endpoints, different endpoints, not only the traditional ones, which are typically survival and global response by the DRC. We are having a very heavy focus in identifying changes in galactomannan index and other biological markers of disease and potentially response to therapy, trying to understand what better outcomes can help us, endpoints can help us to identify a better outcome with a combination therapy. Interesting is the combination therapy will be given during the entire duration of the antifungal treatment. And as I mentioned before, we are right now recruiting centers in the US and EU. In addition to this, still in aspergillosis, the Fury study that we have right now is a phase 3 open label study that is intended for patients with refractory or intolerant to other therapies, so it's a salvage therapy study. Right now it's focusing Candida infections. However, we are about to amend the protocol, allowing also the use in Aspergillus infections as a salvage therapy. We have right now sites for this study open in the US, Germany, Austria, Netherlands, the UK, and Spain. So what are we doing in aspergillosis, in candidiasis? So the Ibrexa has activity, a broad activity against most of the Candida strains that affect the humans, and particularly relevant is, as I was mentioning, that seems to be less commonly affected by FKS mutations that are commonly seen in most of cases. So for instance, in this study, 629 and 663 mutations, you can see here that Ibrexa continues behaving as similar to the wild type when compared to the other candins. The other candins seems to be more commonly affected by these mutations. Also, there are some mutations in the FKA gene that also affects the performance of Ibrexa, particularly 659. So that particular seems to be affecting us as well, but others doesn't seem to have such a profound effect on the activity of Ibrexa. It also has activity against Candida auris, and thank you to many on the audience here generating this data. And it was quite interesting to see that within Candida auris, our activity remains even for those isolates that seem to have high MICs for the candins. Morphologically, it has very profound effects. This is particular in Candida auris that you can see here the effect in the scanning electron microscopy and transmission electron microscopy. So it does something to the bug when it is exposed to. We conducted an MSG study 10 in invasive candidiasis, was a proof of concept PK study basically, and from this we learned that what was the dose that is needed in order to achieve the target exposure from this study. Many of you participated in this study, and thank you for that. We also conducted a phase 2 study in vulvovaginal candidiasis, and from this study we observed that 078 in comparison to fluconazole had at least as good activity as fluconazole in acute VVC, and actually improved activity if you kind of at least numerically better activity at day 25 in terms of improvement in signs and symptoms and mycological eradication, and we're moving ahead in the phase 3 program with this data. So in summary, we have right now ongoing clinical trials. We call it SCYNERGIA, which is a phase 2 double-blind study in patients with invasive pulmonary aspergillosis. This is Ibrexa in combination with voriconazole. We have the FURY study, which is a phase 3 study as a salvage therapy for candida infections and soon to be for aspergillosis as well. And we have the CARES study. The CARES study is a phase 3 study open label in patients with Candida auris infections, and we have it in the United States and in India. In the United States, we're opening that only as an emergency type of protocol. If someone has a patient with Candida auris that would like to have this protocol in their institution, we are ready to start a study in 3 to 5 days. So depending on, we are really ready to really implement those studies very rapidly. So that, I think that's it, and with this, I thank you for all your attention to this.
Thank you. I also like to thank David and Synexus for their support of the MSG through MSG 10 and their willingness to really explore areas that have not been very well explored, especially Candida auris and some of the multi-drug resistant Candida species. Okay. Is Rolando Vianney here? Okay. Hey. Okay. So Rolando Vianney is from Sidero. Come up. Thank you. Thank you.
Yes. Thank you. Absolutely.
Okay. Good evening, everyone. I would like to thank the organizers for their kind invitation to present data, preclinical as well as clinical data on rezafungin. These are my disclosures. SCYNERGIA Therapeutics is located in San Diego. I recently joined them about 4 months ago or so, after spending most of my career in the HIV and hepatitis C world. But we are committed to infectious disease drug development, in particular to the immunocompromised. So this is the SCYNERGIA pipeline. We have rezafungin intravenous for the treatment of candidemia and invasive candidiasis. We recently ended STRIVE, which is the phase 2b trial, that enabled us to move forward with RESTORE, which is the phase 3 pivotal trial. We have already a site that is ready to enroll, the Topolman site, and I really appreciate their commitment. And we'll soon be enrolling patients on the on the treatment study. There's another antifungal prophylaxis in hematologic stem cell transplant patients, the RESPECT study, and that's about to begin enrollment as well. We're in fine-tweaking negotiations with the FDA and EMA about endpoints. There is also a formulation, a sub-Q formulation of resafungin that is, we will start a phase 1 study later this year. And there is the Cloudbreak immunotherapy platform that I won't touch at this time. Resafungin is a better echinocandin, better antifungal. There's a structural modification of the ring here. You can see the addition of choline moiety that allows for a chemical and biological stability of the molecule. So it's designed for a prolonged exposure, a once a week intravenous injection, as well as very high exposures with potential efficacy enhancement against Candida and Aspergillosis. And also the stability of the molecule allows for potential improvement in safety, as well as different formulations, not only IV, but also sub-Q formulations. So the resafungin is widely distributed in body fluids. It's about 4-fold higher than in a plasma, particularly in liver, spleen, and kidney, as well as lungs. Here you can see this is a mouse model of single-dose resafungin. You can see the epithelial lining fluid with very high concentrations of resafungin. And you can see in light, in light blue. One back. Compared to the plasma concentrations in red, and just to suffice to mention, the plasma concentrations are total, as opposed to the lining fluid concentration, which are just free drug. And as you know, resafungin is 97% protein bound. So that's an advantage, as well as a prolonged half-life of 133 hours with very low clearance of the drug. To the right, you can see the mouse liver infection model. In red is at the concentrations of drug in the abscess and necrotic tissue. In light blue are the concentrations surrounding those tissues. And you can see compared to micafungin, about 3-fold higher concentration of resafungin after 48 and 72 hours of single-dose resafungin compared to 1 or 2 doses of micafungin. This is the outline of STRIVE Part A, a study in candidemia and invasive candidiasis. There were 2 doses of resafungin, 400 milligrams followed by 400 weekly, allowing for 2 to 4 doses, particularly invasive candidiasis, compared to 400 loading dose, followed by 200 weekly, allowing to 2 to 4 doses, compared to caspofungin, standard dose. So this is a double-blind study. The primary endpoint was mycological and clinical response at day 14, with an additional mycological and clinical response at day 28 for patients with invasive candidiasis only. So as you can see in the summary of adverse events, treatment-emergent adverse events was very ubiquitous. This is a very ill population. About 80 to 90% of patients had any adverse event. Their study drug-related treatment-emergent adverse events, about 11 to 16% across the 3 arms. Serious adverse event, 13 to 18% also. Very uniform on the 3 arms. There was one in serious adverse event in the resafungin arm and one in the caspofungin arm, both resolved. In terms of the overall response and PI assessment of clinical response at day 14 in the MITT population, we observed in the first column, you can see lower success rate, 57% compared to 70 and 64% in the other 2 columns. But we found out that there were so many indeterminate response, in that particular arm, and those were because of missing patient data or missing evaluations. When we remove the indeterminate response, we exclude those. The success rate was in the 73, 78%, very similar across along all those arms. In terms of overall response and PI assessment of clinical response on day 14, this is an MITT population. You can see as well similar situations. There's some indeterminates there that were related to missing data. When we remove those, we have even success rate. In terms of mycologic response in candidemia and overall response in invasive candidiasis, very high response, 70% or so. Again, several indeterminates. In this case, there were patients that had missing culture data to prove mycologic efficacy. And then when we exclude those indeterminate response, about 77%, 73%, very similar across all arms. In terms of overall success in invasive candidiasis, which is the niche where rifampicin may be more efficacious than other echinocandins, you can see, even though the numbers are small, you can see very good response rate as well. Yeah, so in terms of rifampicin PK/PD, dose selection and target attainment, the target AUC over MIC depicted in red at the bottom of the slides show that caspofungin target attainment kind of in the 50% or so to the right, with rifampicin inhibitor at the lower dose, very high target attainment. This is the RESTORE, the phase 3 pivotal trial in candidemia and invasive candidiasis with 2 arms. This is a double-blind, double dummy, rifampicin versus caspofungin at the doses picked here. There is a global response at day 14. This is an EMA endpoint, as well as all-cause mortality at day 28. We will enroll about 200 patients or so. In terms of antifungal prophylaxis, we have, as you know, hematologic stem cell transplant patients require multiple azoles with drug-drug interactions, overlapping toxicity, as well as pneumocystis prophylaxis with high toxicity as well. We have the potential of using rifampicin with a simplified drug regimen. And these are the 2 arms that we have. This is a phase 3 pivotal study, rifampicin versus a comparator, which is an azole and as well as Bactrim prophylaxis. This is a double-blind, double-dummy study. Primary endpoint is a 90-day fungal-free survival. This is powered for non-inferiority with about 480 patients. There's also, to finish, there are several phase 1 studies, drug-drug interaction studies with different drugs that are metabolized by and can be substrates or perpetrators of interactions with cytokine P450 as well as P-glycoprotein and transporter proteins. And we saw no change in exposure with rifampicin when compared to all the drugs studied. Thank you very much.
Thank you. Very interesting compound. Very interesting compound. Vical. Okay. Yep. Another San Diego-based company, correct?
Correct.
Okay.
Must be something in the water. Thank you very much for the invitation. I'm going to present on our VL2397 antifungal drug for treatment of invasive pulmonary fungal infections. But before I launch into that, let's see. I have a disclaimer for Safe Harbor for forward-looking statements, and I am an employee of Vical. So VL2397, it's a novel antifungal drug in that it's a cyclic hexapeptide that has 3 ornithines that are hydroxymated, and they form a coordination complex with aluminum. This molecule resembles a siderophore, and consequently it gets into fungus using CYT1, which is siderophore iron transport 1 molecule. We are developing this for treatment of invasive fungal infections. And one important thing, too, is that this molecule, because it's unique, it doesn't really interact with cytochrome P450, so there's no potential for drug-drug interaction with this molecule. And one point that's not up here too is it's also very fast acting, especially compared to echinocandins and azoles in inhibiting fungal infection. So the molecule is active against Aspergillosis species, Fumigatus, Pteris, Flavus, and Nigellans, and we're in immunocompromised as well as older patients. It's also active in some Candida species, Candida glabrata and Candida kefir, and then also other fungal pathogens, Fusarium and Trichosporin. And susceptibility is defined by an MIC of less than 2 microgram per mil. So for clinical development, we are collaborating with MSGERC. We also have a potential expedited development pathway. We have, because this is a QIDP designated molecule, we have a very interactive relationship with FDA in designing our phase 2 trial that I'll talk about in a minute. And this trial, the phase 2 trial, is a pivotal trial, and if we reach our clinical endpoints, this would then be available for commercial manufacture. We've completed a phase 1 study using dose escalation, both from 3 mg to 1,200 mg. We've also done multi-dose escalation for 300, 600, and 1,200 mg, and finally we did a 28-day cohort receiving 900 mg daily as well for 7 days, followed by 21 days of 600 milligram. And it was safe and well tolerated in healthy volunteers. Our PK data showed that there was no drug accumulation. By the way, our preclinical data showed that this drug actually gets secreted about 97.5% intact in urine and feces, so there's no accumulation there. The PK/PD analysis from our phase 1 study identified a 600 mg dose for treatment in our phase 2 study. So the phase 2 study, it's a global multi-center randomized open label study. We have 40 major cancer and transplantation centers in North America, Europe, and Asia. So far, the sites that are activated are in the US, Canada, and Belgium. The study will enroll 200 subjects, AML or ALL, or also allo HCT recipients. It's a 2-to-1 randomization, and the comparator, it's going to be physician's choice of VORI, ISA, or liposomal amphotericin. We have a 6-week antifungal treatment, 4 weeks of VL2397 with 2 weeks of comparator. Primary endpoints are all-cause mortality at 4 weeks with a key secondary endpoint of all-cause mortality at 6 weeks, and it's a non-inferiority design. And that's in a snapshot. We do have a poster talking about mechanism of action, as well as we have a clinical meeting on Thursday after the Aspergillosis section.
Thank you. From Vimet to do the next presentation would say, while he's getting up here, that so the VICAL study is the Aspergillosis study is MSG 17. MSG 17.
And we are not from San Diego. So I think most of you heard the Viamet pitch that we're a metalloenzyme drug discovery and development company, focuses equally on selectivity and potency. And just to emphasize why we focus on selectivity, obviously we want to build safer drugs, but we really want to build drugs that have a greater therapeutic index. We feel like a lot of drugs leave a lot on the table because they can't reach levels that achieve maximum efficacy, and that's our goal. So our successes to date have been VT1161, which is now in phase 3 for RVVC under Mycovia, and VT464, which is being progressed by endocrine, that's now in phase 2 for prostate and breast cancers. And we now have 2 robust discovery programs in other therapeutic areas, and we are operating now under Salinity Therapeutics. I would be happy to explain all that to you. I should mention that I tried to explain that to David Bouwar, and he walked away, so it's a little complicated. But most of the talk I will be focused on 1598. This is all preclinical data. So 1598 is a novel oral antifungal for invasive fungal infections. It was rationally designed for exquisite selectivity while retaining high potency. Broad spectrum antifungal, both in vitro and in vivo preclinical data. Excellent safety, preclinical safety. That sets us up for a very high therapeutic window that we feel like we can attain maximum efficacy in the clinic. The IND has been filed and accepted by the FDA, and I think most know that we are focused initially and right now solely on the clinical development for Cryptococcal meningitis, and that CM has been added to the list of neglected diseases by the FDA and is therefore eligible for the PRV, and a lot of you in the audience has helped push that forward. We are in advanced discussions with the DMID folks to conduct the phase 1 study, and we are actively exploring many options to be able to do phase 2 and 3. So very briefly, this is in vitro data that was collected by Nathan. This is about a quarter of the isolates and a quarter of the species that were studied, and this was published last year. And you can see very, very high potency against, in this case, against albicans and neoformans, fumigatus, and cocci. These are the highlight selectivity data. This is against 4 of the liver metabolizing CYPs and a couple of endocrine CYPs. You can see that 1598 really doesn't inhibit these enzymes. Equally importantly is the fact that in vitro IC50s very accurately predict DDIs in the clinic, and so we are very confident that 1598 will not have DDIs, ones that are resulting from inhibition of CYPs at least. And just to present one in vivo study, this is a Cryptococcal meningitis study, a tail vein injection using H99, 6-day treatment, one starting 24 hours after inoculation, fungal burden measured 24 hours after the last dose, and also a survival study, 14 days of treatment followed by a 6-day drug washout. Positive controls with fluconazole and ampisome. Study was done at Evotech in the UK, and it was published earlier this year. So the highlight data is in the chronic, in the acute study. You see the level of fungal burden in the brain at the start of treatment. This clearly expands after 6 days of vehicle treatment. and you can see a dose-dependent decrease reaching 6 logs reduction with 1598 treatment, and then the positive controls of amplisome and fluconazole. And to point out that the mid and high dose of 1598 reduce fungal burden below the input level. And to point out for the 14-day study, for the mid and high dose, the fungal burden reduction is maintained after drug washout. So to summarize, 1598 has demonstrated highly significant antifungal reductions in a number of studies. Several of them have been published. The maximum effects observed in some of these studies were consistent with a fungicidal-like activity, a very, very high level of reduction, reduction of burden below the started treatment level, and sustained reductions even after washout of drug. And just to finish with the fact that in all cases, the highly significant reduction occur with plasma levels of 1 to 5 micrograms per mil. The greater activity, which is really the hallmark of our discovery program, is at levels of 5 to 20. And it's interesting to note that, I'm sure everyone here in the room knows that this is the level that the current azoles sit at. And they have very nice antifungal activity, but we really feel like there could be greater activity if you can go to higher levels. Thank you.
To introduce our next speaker, John Rex, who was at one time one of the key investigators in the MSG. And then he had a turn in career in the early 2000s and joined pharma on the bacterial side, and finally found his way back home and is now the medical director, chief medical officer of F2G, and he's going to tell us in a few minutes about what he's up to.
Great. Thanks, Pete, and it's fun to be back here. Is there somebody working the computer somewhere behind the screen? Okay, because there's a slide with a video that I'm going to need some help with. So let me tell you about F2G's compound, olorifim, and that's our new name. It used to have a number, now it's got a name. Sounds like an Italian opera, olorifim. So olorifim is the first erotomide antifungal. It inhibits DHODH, an enzyme that's involved in pyrimidine biosynthesis. Every living being has this enzyme, but they're not all the same. The human enzyme is dramatically different than the fungal enzyme, 2,000-fold difference in IC50. Activity follows enzyme structure. We actually can predict activity better from phylogeny than from doing MIC, so they're the same, really. And it's active against all the Ascomycete septate molds. It's not active against things that aren't Ascomycete septate molds, so Candida, Crypto, Mucor. There's no cross-resistance with the azoles, the echinocannins, the polyenes, or flucytosines. When it's active, the MICs are consistently similar to that for Aspergillus, which is tightly clustered around 0.03 to 0.06 milligrams per liter. For Aspergillus, it's the same MIC for Fumigatus as it is for pick your favorite cryptic species. Similar ICs for the other Ascomycetes, Scedosporium, Laminospora, and the endemic dimorphs. One that's inconsistent on is Fusarium. We're not sure what that means yet. We're working on that one. So next slide. So it stops growth quickly and cell rupture follows. The person behind the screen, would you click on the little picture in the upper right if you're listening? I'm hoping someone's, there we go. Okay, so that video is going to play in a loop. And at time 0, what you have is a growth tip of Aspergillus fumigatus under the microscope, and we drop in, it's 16 hours old, and it's buzzing along doing its thing. And we drop in olorifim at 0.1 mg per liter. And you'll see it slows down, and it stops. And by about an hour, no more growing going on. And if you wait a little longer and look at it by transmission electron microscopy, what you find is dead fungus. So I have 2 pictures there. On the lower left, it's a normal hypha. Didn't touch anything. Ignore the little gaps, the little white spaces, that's preparation artifact, but notice the nice normal nucleus, the nice even cell wall. Look on the right where you see thinned, broken cell walls. You see nothing inside the cell. You see a weird, distorted form on the right. You can actually watch them pop under the microscope. It's kind of fun. All right, next slide. All right, so we can produce adequate levels in man, both PO and IV. What's adequate? Well, we've worked extensively with William Hope on the question of what is adequate, and the PD driver shows that it's a Semen-driven drug with levels around 0.1 to 0.3 milligrams per liter being enough. The in vivo activity really matches the in vitro activity. We can document it lots of different ways. And in man, it's PO, it's IV, it's linear, it's got predictable DDI. It's cleared by a lot of CYPs. It doesn't induce any CYPs. It's got very weak 3A4 inhibition. It's gentler than fluconazole. You can combine it with tacrolimus and the other things like that. Finally, phase 2B trial is now underway. I think I've said this stuff at the top: novel mode, lethal, dose proportional PK, tolerability. The open label salvage study is open now for subjects with proven invasive fungal infections or probable invasive aspergillosis due to any susceptible genus or species, and limited treatment alternatives. Our particular focus is Aspergillus, Scedosporium, and Laminospora, but we are casting quite a broad net. Sites are opening globally, other studies to follow. Thank you very much for your attention.
Next speaker is Sean Bauman. Where's Sean? Here you are, from Emmy, and we're going to change the pace a little bit here. This is, Emmy is our first diagnostic company that'll be presenting this evening.
First, thank you to Pete and the organizers for putting together such a great meeting as usual. So Emmy's a diagnostic manufacturing company that was founded in 1979 in Oklahoma. So we've been at this for quite some time. And there we go. Over 70 products, 60 plus countries across the globe. Really our mission is trying to push diagnostics close or further down the healthcare chain. Many times they're happening in reference laboratories with turnaround times that are often unacceptable. And ideally, we'd like to push them down to the clinic level where that makes sense. Tell you a story about Cryptococcus. So the current director of CDC, when he was at the University of Maryland, Robert Redfield, gave me a call one day and said, "Hey, Sean, we got crypto patients that are dying all over the place in Africa. Let's do something about it." And naively, I said, "Great, tell me what I need to do." And he said, "Get on a plane and go to Mwanza, Tanzania, and teach people about the state-of-the-art crypto diagnostics." I said, "Great, I'll go. I've never been. I don't know how to even get there." I knew that I probably couldn't carry a bunch of liquid in my suitcase, so I shipped this kit, a latex agglutinin kit, agglutination kit in advance, and that's how it showed up in Mwanza. That's not how it left my facility. I didn't even know how to ship a product to Africa or to to get a product in those types of locations. Saw a picture in Kenya, took a picture of this sign. It's "Keep it simple," and it's really changed the focus of our company. And those of you who've ever seen a latex agglutination test, it's pretty simple by most standards, but it's really hard when you don't have electricity and freezers and refrigerators and good shipping companies. And although they do have FedEx motorcycles in Tanzania, that's just doesn't keep the cold chain nearly as well. We came up with this dipstick test, the CRAG LFA. It's very, very sensitive and specific. Some consider it the new gold standard. It's been the basis of a WHO guidelines that now it's in its second revision. Lots and lots of partners have helped make that happen. I will speed up. There we go. Now there's 27 countries that have CRAG screening in their guidelines, and that's in a very, very short period of time. So we're trying to do similar things. I'm going to skip over this crypto if I can. We have a new crypto test that will basically segment patients based on a finger stick into high probability of having cryptococcal meningitis and a lower probability of having cryptococcal meningitis. Well, we turned our attention to aspergillosis, really seeing the problem is many places have, they send out for testing, have long turnaround times. So time to result in days. We built a little galactomannan lateral flow assay that's now CE marked for serum and BAL. So now the whole idea is you can get time to result in minutes. And you'll see this theme happening over and over. For Coxy, it's typically a reference laboratory type test. Time to results in days to weeks, depending on how close you are. We build an LFA detecting Coxy antibodies, turnaround time in minutes. Again, very good performance. We even have a lateral flow assay that'll tell you IgG or IgM for Coxy. Histoplasma. Traditionally, it's been a reference laboratory test. Again, longer turnaround times and have limited availability outside the US. Now we have a histoplasma ELISA that has availability with worldwide, and I believe Anna will be speaking about that on Thursday. Time to result, again, in hours. So one last slide, the most important one. Acknowledgements. People all over the world have contributed to all this work, and there's some that aren't on there. One last not so bold prediction. I've done this at every MSG, and I'm going to do it one more time. So thanks for agreeing, Pete.
Too much time in the lab, Sean. Too many hallucinogens. Okay. Our next presentation is from T2 Diagnostics, and that will be Sandy Estrada. I'll say, we have worked pretty closely with Emmy and T2 in the development of some of their assays, so looking forward to this update. Thank you, Sandy.
Thank you so much. Again, thank you all for allowing us to be here tonight. So I'm Sandy Estrada, the Director of Clinical Affairs for T2 Biosystems. Also new to the position. I've been with T2 for about 3 whole months now, but well aware of the great collaboration of many of you in this room with T2, so thank you very much for that. So sorry about that. It's been a long day. I traveled from South Florida today. So as many of you are aware, T2 Biosystems has a new platform of technology in the rapid diagnostic space in that we're detecting sepsis-causing pathogens directly from whole blood. And so this is the instrument over here on the left. Then over on the right, our T2 Canada assay, which has been available commercially for about 4 years now, detecting the 5 species of Canada that you can see on the screen or that you did see on the screen. And our newest product that we won't discuss tonight, but very excited to announce that we did get FDA approval of our T2 bacteria panel in May of this year. So really what makes the difference for T2 Canada compared to traditional culture, we are able to go directly from whole blood and 3 to 5 hours later have an answer and detect greater than 90% of the species that are out there compared to 1 to 5 days for traditional blood culture with about 50 to 65% detected. And of course, compared to other molecular diagnostics that are out there, also with great sensitivities and specificities, but dependent on post-blood culture technology compared to pre-blood culture. So as the years have gone on, there have been more cases reported demonstrating T2 Canada is more sensitive for deep-seated candidiasis compared to blood culture-based methods. So data coming out of Henry Ford from 2016 showing 11 T2 positive, blood culture negative, but culture positive from non-blood specimens, as you can see listed there. Also 2 cases of Candida in Paima where T2 Canada detected but blood culture missed, and 1 case of polyfungemia where all species detected by T2 Canada were also confirmed by blood culture. You can see similar data from other institutions that I won't go into at this point. So additionally, in addition to the sensitivity, specificity, and ability to find these infections, the real focus is how can this impact the management of candidemia. So again, in the data from Henry Ford, published in a Journal of Antimicrobial Stewardship last year, T2MR was shown to lead to a 7-fold reduction in time to appropriate therapy of 5 hours versus 39 hours prior to implementation of the technology. The median time to appropriate antifungal therapy was reduced from 39 hours to 22 hours once it was integrated into the Antimicrobial Stewardship Program. And Canada was definitively identified in 93% of patients after implementation compared to 57% prior. Additionally, one finding that we hadn't really considered but that Henry Ford looked at was the reduction of Candida ocular involvement from 30% to 12% after implementing T2MR, again, likely due to the more rapid diagnosis and treatment of these patients. Whoops. Sorry, I'm just struggling tonight. So some of this data on the left is actually from my former institution, Lee Health in Fort Myers, Florida. We were able to implement T2 Canada as part of our antimicrobial stewardship program there about 3 years ago. We also saw a significant decrease in time to appropriate therapy from 34 hours to 6 hours in our candidemic patients. We were able to avoid empiric antifungal therapy in over half of T2 Canada negative patients. So again, another antimicrobial stewardship impact, resulting in about $280 of cost savings per tested patient from these antifungal costs, which is helpful in justifying implementation of new technologies for our institutions. On the right-hand side, we see data from UPMC, also showing significant duration of therapy reduction and reduction in antifungal expenditures reduced by 47% after the implementation of T2 Canada. So again, I think the main point that we have being on the market for a few years now is just the increasing data of really how the real world evidence is adding up for the implementation of T2 Canada and various institutions and utilization along with antimicrobial stewardship programs. As far as what's coming next, we do have some gram-negative resistance panels being developed as well as an RUO Candida auris assay. So thank you so much for your time.
Our next presenter is Dennis Hooper from Real Time, and he's going to go through the data of this new assay. Where's Dennis? Dennis? Okay. MicroDart, PCR.
Thank you. My name is Dennis Hooper. I'm one of the founders of Real Time Lab and MicroDart PCR work that we do in Dallas. I'm presenting here the early and rapid diagnosis of fungal infections that we have developed in the last few years. You all are very used to seeing this in the clinically significant fungal infections in invasive candidiasis, invasive aspergillosis, and mucormycosis, and the clinical management is hampered by all the difficulties in diagnosis, and that's what we're going to talk about. First of all, I'm a little out of breath because we came all the way from Dallas and we started with American Airlines. Now, if you ever fly through Dallas and you know American, it's always late. Well, always, especially when I'm on the flight. So, hence we just walked in. Whew. So, we're going to talk about the diagnostic approach of these invasive fungal infections. All of these things you're used to seeing, the specificity and sensitivity of the current tests are very limited. The negative predictive values in some tests delay therapy in patients with the infectious fungal infections, and there's multiple tests that require adequate logistics in support. The culture method is about 50% sensitivity and 50% specific, as you all know, but we have to talk about this because it's the gold standard. We have met with FDA concerning this, and it is a concern that you probably all have as well as we do. The beta-D-glucan is the same thing that you can see here. The PCR method is the one that we're most concerned with at our company. This is 90% sensitive and 100% specific, but has a very high accuracy but low commercial availability for various reasons. We also know of the galactomannan, the mannan, and a combination of all these, showing 87 to 84% sensitivity depending on how you take the literature and combine it. We know the problem, as you all are aware, of over the 50,000 patients that undergo transplant surgery and bone marrow and solid organ work. And we know the need There's a fast and accurate method to identify the nature of the patient's fungal infection, and we at Advatech in real time have the solution. Advatech Diagnostics is an LLC located in Dallas. It has developed patented DNA probes for PCR, which determine specific fungal species in patients' blood within 4 to 6 hours. Its probes have been analytically validated, and we have 98% sensitive and 96 to 98% specific. The competition, as you see and you've heard tonight, the blood draw is taken and put in a blood culture. We wait for it to grow, and then we can do a antibiotic sensitivity as well as a ID. We can also use other diagnostic products such as T2 and others that are present in this room using the blood culture and do a susceptibility and then identification as well. The bottom one is what Advatech has. We have a blood draw and we have a candida identification with 4 to 6 hours. We can save time, money, and lives. Our devices are Aspergillosis. We have Fumigatus, Flavus, Niger, and Terreus. We also have Candida albicans, Glabrata, Krusei, Tropicalis, Parapsilosis, and now we have Auris. The pipeline that we have developed is methods and compositions for identifying and quantifying the microbial DNA by doing a dual amplification, hence the name MycoDART. Mycology, DART, dual amplification, real-time PCR. We also have multiple patents on mycotoxin identification, and these are, some of them have been issued and some of them are pending. We've done studies with Tom Patterson on Aspergillus fumigatus in guinea pig. We've also done studies with Tom Patterson as well, looking at Candida in mice. Our analytes detected I've listed here, and our matrix is whole blood, plasma serum, and BALs. We've done a study with the University of Florida at Gainesville, and we've got those samples, and we also have listed in our assays of validation that we can detect down to 10 CFUs, but more importantly, we report limit of detection in copies per microliter of blood. Summary of our studies are Aspergillus, Candida, and we'll have this available for you to look at over these next few days. And then these are our studies that we have done comparing T2 and our MycoDART and using our spike samples. So we're here tonight to say that we came all this way from Dallas to meet all of you and to also talk about our MycoDART presentation. We're doing this with different study groups throughout the United States. We've signed up 3 of the 6 in signing contracts, and we've started collecting specimens from Candida patients in the preclinical trial or study. So we thank you again for letting us come and meet you all. We anticipate having you discuss with us what you think we should look at in the future and in the next few months. And we intend to publish this data, and we encourage your input. Thank you.
Thank you very much, Dennis. Our next to the last speaker will be Mick Hannon. Is Mick here? Oh, here we are. Okay, Mick Hannon from Mycomed. You guys are in Baltimore? Okay, all right. And Mick has worked, I believe, closely with Karen Marr.
That's
Okay,
right.
good. Welcome.
Thank you. Well, thank you for having me. So I'll start off by just saying I'm the Chief Business Officer for Mycomed. Mycomed is a fungal diagnostic company. We were spun out of Johns Hopkins in 2014 and actually currently reside in the incubator, Johns Hopkins incubator office called Fast Forward. Our first product is a product to detect urinary, through urinary LFA, Aspergillosis, and we have several additional programs in development. We have a broad pattern of intellectual property, and to date we've been funded by basically state and federal grants. As was said earlier, Dr. Karen Marr is actually the leader of our company. She's the executive director and CEO, and the technology is based on her 20 years of experience. We are working on a couple different things, but most particularly the diagnostic for Aspergillosis. There are two key discoveries that are important there. The first is an antibody that we call 476, and then also a process on how to eliminate an inhibitor to the reaction in urine. 476 is a monoclonal antibody produced by Aspergillus spores. It detects a galactomannan-like antigens, is what we originally called it, with high affinity. It's now known to be short-chain galactofuranose on small molecular weight glycoproteins. Galactofuranose is only made by non-mammalian eukaryotes and some prokaryotes. And those small glycoproteins have rapid localization into urine in affected animals. Also, as part of this, we discovered a process to eliminate a naturally occurring inhibitor of the antigen-antibody binding. This is some data that we presented earlier this year, which was published in Clinical Infectious Disease. This is using what was then our optimized prototype. It has analytic sensitivity to 150 to 200 nanograms per ml. It's a visual read, and it's semi-quantitative. If you can see on the picture here, you can see as the antigen concentration goes up, so does the strength of the line on the test. We did 2 clinical studies at Johns Hopkins, who were diagnosed per EOR, TC, and MSG guidelines with 78 patients. In those people who had clear diagnoses, we had an 80% sensitivity for proven or probable IA, and we had a 92% specificity in patients at risk but not IA. Those 2 patients that were false positive actually had histo. In hematologic patients, we have a greater sensitivity at 89.5, and the specificity remained the same. Importantly, in people with possible IA, unclear diagnosis, our test was able to pick it up in 38% of them. And in those folks who have no disease, they were all negative. The healthy volunteers, they were all negative. And we have a poster here at this session, poster number 18, which has updated data on these studies. We are on our course to get to a 510K, and we had a pre-510K meeting held in collaboration with both Aztec and John Wingard's group, and with Chris Benson, who many of you may know was instrumental in getting the Platelia assay onto the market. Our studies are going to be based on urine samples from NIH, Aztec contract, as well as from JHU. We have performance and reproducibility studies being done at 3 clinical labs, Hopkins, Duke, and Pitt. We have bank samples to determine sensitivity both in the pediatric and the adult population. And we are doing specificity studies using banked and freshly collected samples. We think that MicroFlow ASP, our product, is going to be a significant advantage over the current methods. We think that our target biomarker galactofuranose is a better product. It is smaller chain and therefore may allow for greater sensitivity. We do it in urine rather than serum or BL. It's a single dipstick test, which is capable of batching. Our turnaround time is 15 minutes, less than 15 minutes, and our sensitivity and specificity rates have been in the high end versus the Platelia test. And the only thing that's really needed equipment-wise is a microcentrifuge, so it allows for greater implementation across clinical settings. And I'll just say that we intend to submit our 510K at the first quarter of next year. Please check out our poster number 18. And if you want to ask me any questions, please catch me in the hallway. Thank you very much.
Our last industry presentation, Susie Fleming from Leadient, will tell us what's going on with Leadient. And I would also say after conclusion of her talk, we're going to have some brief summary remarks, and then we'll be adjourned. Promise.
Absolutely. Okay, let's make sure I have this turned the right way. Thank you all for your, I should have asked for espresso shots to be passed out right before, so I stand between you and the rest of the evening. I thank you all for having us, Leadiant. This is the first time that we've been here at this presentation, and I'm here with my colleague, Dr. Mark Resnick, as well. So just to give you a little background about Leadiant Biosciences, we're hoping that we can make a commitment to a strong future. We're built on a legacy of rare disease. So many of you may have heard of Sigma Tau as we were before, but we're now Leadiant Biosciences. So if you haven't heard of us, hopefully you will pretty soon. We're leadership that's based on rare disease therapies through innovation and excellence, and that's where we have tried to put our foundation as well. Our corporate strategy is to see opportunities to improve care and bring novel and effective therapies to the market and those that are affected by rare diseases. So our diseases span anywhere from disease states with less than 40 in North America to up and dealing with, so we have ABLSET for invasive fungal infections, but we kind of run the gamut when it deals to a rare space. So this is actually one of our patients with ADA SCID, and we really want to make a commitment, and if you know many of us, you'll find that we're extremely passionate. We love what we do, and we really want to partner with those that we can, especially in the communities, and try to find those areas that we can bring as much value as we can. So just a little bit about Leadiant. We're a U.S. wholly owned subsidiary. We're based out of Gaithersburg, Maryland. Our main office is based in Italy, and we're the sister company also as well to our UK subsidiary. We're solely focused on rare diseases, and we currently have about 8 marketed products. And like I said, they kind of run the gamut in various disease states. But we do have a robust research and development, and we're trying to keep that on a global perspective as well. And there's about 60 of us, so we've been around for about 20 years. As I said, we were previously Sigma Tau. We've been Leadiant for about a year. So if you're not familiar with Leadiant, that name is fairly new to a lot of people. So as we come into facilities, it's a little new introduction there. And again, just to reiterate, our mission is that, we work with the rare disease space. We love working with people who are as passionate as we are in the communities. And we hope that we're a resource as well, because anytime if you've worked in the rare disease space and you know these families are incredibly passionate, and we feel the same. We absolutely love being involved in this. So thank you again for letting us be a part of this, and we hope that you enjoy this conference and we get to spend a little more time with you and getting to know you. Thanks again.
If we can bring my slides up, we're gonna wrap it up in a few minutes. But before we do that, I want to make a couple of observations. First of all, to our industry sponsors and to everybody in the room, it's obvious that each of you could have spoken for an hour about your data, about your products and your vision, and there's just no way to fit that in. So we recognize that you're getting short shrift on this. However, one of the purposes of this meeting, because this meeting is interactive, the main reason to have you here and speaking is so that people can see you, and over the next 2 days, begin to develop relationships, develop connections. So this meeting is about connectedness. It's about forming collaborative bonds. So I would encourage all the investigators here and the people, whether they're clinical laboratory, to reach out and meet some of the sponsors, those that you have a real interest in, and start trying to develop some of those relationships. The other observation I want to make is really the obvious one, and I'll just point to John Rex and say, John, can you imagine in 2002 that would be dealing with this many new products, this many new diagnostics as we are now? I mean, it's unbelievable. Those of us who have been in this arena for a while, and there are a bunch of us here that it's unbelievable that we're dealing with, you know, no less than 10 new products and a host of diagnostics that that are just really kind of yet to find a place and yet to be studied. So this is all really, really great news for the world of mycology, and one of the reasons we have this meeting. So thank you all again. Thanks, industry, for what you do for us.